He is head of the National Bioenergy Laboratory at the National Renewable Energy Lab, and I'm really excited that he's here to talk to us today about something that I think is on everyone's mind in the biodiesel community, which is feedstock. Everyone knows that it works with biodiesel that feedstock's a growing issue, and both on the small and the large scale, and it will continue to be an issue without innovative solutions. And these are the solutions that NREL is working on, and I'm excited to have Al. Welcome.
Well, thank you very much, Jonathan, for giving me the opportunity to speak here today. So the title of my talk today is The Promises and Challenges, and I think we have to understand both of those words, of algal biofuels, because some people think that we're already here, but we're actually a little bit further away than we think. Another potential title for this talk is It's Not Easy Being Green, although it is getting easier to be green these days. So what I'm going to do today for the next hour or so is going to give you, first of all, an introduction and a rationale for why we should be developing microalgal-derived biofuels, a review of previous work at NREL, the Aquatic Species Program, and some of the accomplishments, who is interested in microalgal feedstocks right now, and then talk a little bit about beyond the Aquatic Species Programs and the types of things that we're currently doing at NREL to develop algal biofuels feedstocks, talk a little bit about an act in 2007, and from that was spawned the Congressional Algae Report, and then a summary and future outlook in terms of the promises and challenges. So most of what we do at the National Bioenergy Center is really to work on cellulosic ethanol. Cellulosic ethanol really addresses the gasoline market. What we're trying to do is address that 140 billion gallon per year addiction that we have to gasoline. However, cellulosic ethanol does not address the need for these higher energy density fuels like some of these that you see over here, like biodiesel, diesel, and jet fuel, which are significantly higher in terms of energy content than ethanol over here. And so what we're dealing with here is U.S. diesel on road is about 44 billion gallons, 20 off, and then jet fuel is about 25 billion gallons per year. That's both combined commercial and military use. So that is a lot of diesel that we have to look to replace, and it's not going to be done with cellulosic ethanol. So obviously everybody knows biodiesel, but there's a huge dilemma, as most everybody knows, with biodiesel these days, and that is that the triglycerides, the oil from these oilseed crops, really cannot come close to meeting the demand that we have currently for the U.S. diesel demand, which is 60+ billion gallons per year. If you were to convert all of the U.S. soybean crop to biodiesel, I mean, it would only replace about 3 billion gallons of that 60+ billion gallons of diesel fuel a year. And then estimated worldwide, that's only about 13 billion gallons. And again, it's just a drop in the bucket. So, you know, the cost of biodiesel feedstock for these producers is increasing. It's probably $4.80 or maybe closer to $5 right now. I don't know, have the exact cost right now. So their input costs are high, and they have to compete with the high-valued food market. So we need to start thinking about alternative sources of oil.
Question?
Yes?
You want questions now or at the end?
Anytime, it's fine.
Okay.
Yep, I'm good.
You just mentioned the soybean crop being converted. If you were going to do that, why would you grow soybean as opposed to rapeseed or something like that? And if you were trying to maximize oil production per acre, what would the crop be, and what percentage could you get?
Oh, that's a really good question. I'm not really too sure. I mean, the predominant feedstock in the United States is soy, but of course, all of that you can't convert into biodiesel. Most of it goes to food.
Right. Now I understand that if you're growing it purely for a feedstock,
right?
What would you grow in? Okay.
So I guess we can maybe touch bases afterwards because I'm not totally understanding the question. So algae, microalgae and macroalgae, i.e. the seaweeds, really have numerous bioenergy routes. For instance, in microalgae, obviously you can produce a variety of intermediates like hydrogen. There's a very robust research group here at Mines and at NREL. Microalgae can also produce lipids and carbohydrates. They can also, even the biomass can be used convert to a variety of fuels. With regard to the lipids, of course, those can be converted into the biodiesel or given to a petroleum refiner, and those can be converted into alkanes or essentially green diesel. Carbohydrates can obviously be used to make ethanol, much like we do with cellulosic ethanol, but they can also be reformed into alkanes and green diesel. And then lastly, the biomass can actually be anaerobically digested to produce methane, or put it through some sort of thermochemical process like gasification, make syngas, CO and hydrogen, and make Fischer-Tropsch liquids, which again, is very akin to a diesel. So, these organisms here really have a nice portfolio, if you will, of intermediates that can be transformed into a variety of energy fuels. Why fuels from algae? Well, first of all, algae produce more lipids per acre than any terrestrial crop that we currently have in cultivation. Potentially anywhere from 10 to 50 times more. Algae, growing algae, we can use marginal, non-arable lands, so you're not going to be growing algae in Iowa where they're growing a lot of corn and soybeans. So you're going to be using areas like the desert Southwest, Arizona, New Mexico, Texas, etc. You can use saline or brackish water, because many of these species actually live in the ocean, live in very high saline environments. The likelihood is that you're probably not going to make this economically, commercially viable using fresh water, because fresh water is going to be at a premium in the future. So having an organism that can use saline or brackish water is going to be a big benefit. There's going to be no competition with food, feed, or fiber, which is good, so we omit that. These organisms can use large waste CO2 vent sources, and so you can co-locate this to, let's say, a coal or natural gas fired power plant and take advantage of the CO2 that is in their natural flue gases. So algae really do, based on some of these things that we have here, have the potential to replace a significant, if not all, of the diesel usage that we have in the United States. Are we there yet? Obviously not. The other thing that in the future we can envision is an algal biorefinery that's not only going to produce the oils for producing fuels, but also protein that could be maybe used as an animal feedstock, or carbohydrates, which also could be used for fuel production as well. Here is a graph that just shows some of the oil yield potential of algae compared to some terrestrial crops. I've highlighted soybean over here, about 48 gallons per acre per year. Some of the higher ones like jatropha or oil palm can approximate up to about 600 gallons per year. And some of the publications have bandied about this number that you see right here. This is 10,000 gallons per acre per year. Now this number is really based on this calculation right here, that is having an algal species that can actually make 50 grams per square meter per day, and that algal species contains about 50% of its dry weight in that oil. That's a pretty optimistic view of things. I don't think we're there yet. During the aquatic species program days, and I'll get into that, the average over an entire year was only about 10 grams per square meter per day at maybe 10 to 20% lipid. And so this, while maybe attainable, we ain't there yet. A more near-term value, I would think, is somewhere in the neighborhood of about 1,000 to 1,200 gallons per acre per year, and then maybe we can aspire up to 5,000 to 6,000 gallons per acre per year, but it's still 10 times more than what's produced by oil palm. This just gives you a little view in terms of what the chemical makeup of the oil is. This just gives you the fatty acid composition of soybean, which is mostly in the C14 to C18 range. And here are just 3 different algal species, a green, a diatom, and Batriococcus, very slow-growing algae. And you can see that there are some minor differences here and there, but overall the content of the fatty acids is not that dramatically different from that we see in soybean. So that's a good thing. Production of algal oils is going to require a number of things that I've alluded, some of which I've already alluded to: land, sunlight, water, CO2, probably from a flue gas someplace, or whether there in the future there are dedicated pipelines that are going to be crisscrossing the United States with CO2 that could pipe into an algae biorefinery, would also be very useful, and a variety of macro and micronutrients, phosphorus and nitrogen and things like that. Here are kind of a visual concept of what are some of the resource requirements for growing algae. So let's just compare soybeans and algae, and we're going to be rather conservative. We're going to say that our species makes about 1,200 gallons per acre per year. We're not going to use that 10,000. And let's say we wanted to make 3 billion gallons of each of these. What is the amount of land that you would be required to make these amounts? If you're talking about soybean, which produces about 48 gallons per acre per year, the total number of acres that you would need is about 62.5 million acres, which is this larger box here, which is the most of Arizona. So that's the land mass that you would need in order to produce that much soybean, or for that much soybean oil. Algae, even at 1,200, would only need about 2.5 million acres, which is that piece of land there. Still a heck of a sizable chunk of land. But you can imagine what happens when you're able to produce more algae that have more oil. This number can go up to 5,000 or 6,000 gallons per acre per year, and significantly reduce even that land mass that you see here. Don't get me wrong, the resource requirements are going to be huge for this technology. Just the water usage alone, we visualize that there could be 6 trillion gallons per year required for this algae farm in order to produce 3 billion gallons. But still, that's on the order of the type of water that people use for cooling various power plants. Yes?
I live in Arizona, and in touch with some of the folks through Polytechnic and XL Renewable. I wonder if you could speak specifically to some of the differences and strategies that they're using to develop algae oils that are less and more water intensive, open ponds versus tubes in the desert.
Right. We can get, there's still a huge controversy between is it better to grow open ponds versus closed systems. I think over the last 12 years there's been some very novel photobioreactor systems that have come online, but the bottom line here is going to be what is the cost. I mean, I've seen some, and I'll show you some very elegant closed bioreactors. They look absolutely fantastic, but during the aquatic species days when that program closed down, their future lookout was the only way we think you're ever going to get this commercialized is with open ponds, because those are the cheapest way that you can go. In fact, some people are even arguing that maybe it's not even open ponds that are cheapest, it's maybe somehow co-locating algal growth with wastewater treatment plants, because all your nutrients are essentially free. So we'll touch a little bit more about that later on in the presentation. Yeah?
How do you use 2,000 gallons per gallon of algae of oil per year? Is that evaporation from the ponds?
If you're talking about an open pond, you could actually be losing about 1 centimeter off the top of that pond a day in terms of water. So there's going to be a huge water requirement. That's why some people are going to closed bioreactors where you reduce that evaporation, maybe don't need as much water, but then again, the cost enters into that photobioreactor system.
Has anyone looked at growing algae in the ocean per se,
no.
Similar to a solar pond?
No, but people have been talking about it, but there is no movement in that direction quite yet.
If you actually go to the U.S. Patent Office and watch how many patents are streaming in on concepts right now.
Yes, huge. It's huge.
The idea, patent.
Yeah, it's huge. And not a lot of issued patents. I'm monitoring that. And over the last 6 months, I don't think I've seen one issued patent in this area, but if you take a look at the ones that are popped up as applications, they're huge. All these companies, these hundreds of companies that are out there.
I don't think there's a precedent yet for the patents. And once they set the precedent, you're going to see them all get issued.
That's right. That's right. Good point. So during the aquatic species days, this was a project at NREL that ran from about 1978 to about 1996. Really focusing in on algal biofuels from oil. And during that time, I mean, the 3 main areas that we focused our studies on were, first of all, collection, screening, and characterization of algal strains from a variety of places around the United States: Arizona, California, New Mexico, Texas, Utah, etc. And they were able to build up about 3,000 algal strains during that period of time. But notice how long it took them. I mean, this was about a 5-year period. I'll tell you a little bit later on now, we have some more high-speed, high-throughput capabilities now to hopefully reduce this number. Then we've looked at the biochemistry and physiology of lipid production. What are the triggers that allow these organisms to produce lipid? And then in the latter days, some very preliminary genetic engineering, development of genetic tools, and trying at least kind of a shot in the dark to see if we could improve oil production by overexpressing a gene in one of these diatom species. But in addition to the lab studies, there were a number of outdoor culture studies that initially first started in wastewater treatment ponds, and then later in California and Hawaii, some smaller 100 square meter ponds, again these open, these raceway type of ponds, and then that terminated in a much longer study at Roswell, New Mexico. Why they picked Roswell, New Mexico, I'll never understand it. Maybe there's some alien connection there, I don't know. But over that, they ran that pond basically for an entire year, and were able to only get about on an average 10 grams per square meter per day, with about a 10 to 15% oil production. On good days, yes, they hit 50 grams per square meter per day, but at night the temperature cools down, and you're not going to get the growth rate that you had. So this was a very, very important study. Now, from that study, it was concluded that there was ample land, water, CO2 resources in the Southwest to make about 30 billion gallons of fuel per year, which is about half of that diesel that we use. The economics, however, were challenging, they thought. Biological productivity is still the largest influence on fuel costs, and my background is molecular biology, genetics, protein engineering, and I'm absolutely convinced that we're going to need a genetically modified organism in order to ultimately get this carried through to commercial extent. Capital costs are also huge. That's why when you look at open ponds, closed ponds, you really have to look at the costs, because those capital costs are huge. The aquatic species also thought that the only way you're going to do this open ponds is with ponds that are unlined. So if you can get an area that has a lot of clay, very hard baked clay, just dig it out and fill it up with water, and away you go. Cheap, cheap, cheap is the way to go here. Land costs, they figured, were minor, although if you're going to be building this on the coast someplace and fighting with resorts and things like that, maybe land isn't going to be as cheap in order to take advantage of seawater or things like that. The program was actually concluded in 1996 because it was not competitive at that time with petroleum fuel. Depending upon how you actually costed the unextracted lipid, it went from anywhere from $59 a barrel to $186 a barrel, and that was nowhere near the $20 or so a barrel of petroleum at the time. And this is an interesting quote. I think this is just amazing. DOE at that time justified shutting this program down because they expected petroleum to remain relatively flat over the next 20 years. And obviously we know that it hasn't. The other reason that they terminated this program was they really wanted to focus more of their efforts on cellulosic ethanol. Yes?
What do you think are the challenges of using a local strain versus genetically engineered?
In fact, one of the recommendations from the aquatic species program was, depending upon where you're going to be setting up this biofarm, it's best to start out with indigenous species, because those are the ones have been accustomed to the light levels, have been accustomed to the water chemistries that are in that water. So at least start with that. But I think long term, we're talking 5, 10 years, maybe even 15, I think you're going to need a genetically modified organism, because you're going to need a way to control the production of that lipid. So during the aquatic species program, as I mentioned, about 3,000 strains were collected. At the termination of the program, only about 300 of these strains were sent on to the University of Hawaii. Those have been kind of quasi-maintained, and to date, maybe only about 150 or so strains remain today. We've gotten actually a few of them from the University of Hawaii. Huge advances in applied biology and design of these algal production systems, running this open pond facility for over 1 year. And then at the termination of the project, they actually came out with this report, which is still being referenced today. It's the Aquatic Species Closeout Report, and you can easily get it on the web as a PDF. Here is the site here, but you can just Google ASP Closeout Report, and you pull up this huge report. It's a starting point for many of the small companies that are coming online these days. So it's really an important resource worldwide. Here is some of the future directions at the time of the closeout. Put less emphasis on outdoor field demonstrations, and here it is, more on the basic biology. We don't critically understand the fundamentals of how these organisms are turning on lipid accumulation and what are the pathways and how are they regulated. More on the biology. Take advantage of plant biology. There's a lot of things that people have been doing in plants over the last number of years that could really benefit this particular area. This is an answer to your question here. Start with what works. You know, use native strains initially because those have been adapted to the local environment. Try to maximize photosynthetic efficiency. Easier said than done. Not an easy thing. People have looked at reducing the antenna size of algae, and maybe they help out a little bit in terms of the shading effects and things like that, but maybe not really. So there's going to be a much larger research effort on maximizing photosynthetic efficiency. And here, this is what I live by. You know, really set realistic expectations for the technology. I literally get several calls a day from people who, you know, they have a pond in their backyard and they want to know if they can make biofuels. Literally, I mean, it's amazing. And then other calls are more serious companies doing due diligence, etc. But this is the framework that we're working in. So set realistic expectations for the technology. Yes.
So for the production, I'm sorry.
No, sorry.
Okay, well, I was just wondering about I'm sorry, I lost my train.
Okay, you can ask me afterwards. Yep. So, what's changed since 1996? You know, this guy. $4.14. You know, crude oil price has seen record highs, although they've fallen recently. Diesel's no longer 60 cents wholesale. You know, it's currently closer to 5, and all the truckers are lamenting this. Greater emphasis on energy security, interest in carbon capture because of all the greenhouse gases that we have going up in the atmosphere. Over that 12-year period, new photobioreactor designs, and I'm hoping that there have been advances in material sciences that will hopefully bring down the cost of some of these closed bioreactors. There's also been an explosion in biotechnology over the last 12 years. We didn't have some of the sophisticated omics technologies that we have now. You know, the transcriptomics, the proteomics, the metabolomics. Those sorts of things are really going to help us focus in on the biology to figure out where the pinch points, where the bottlenecks in these particular metabolic pathways.
Question.
Yes.
You talked about the CO2 capture on there, and you mentioned earlier from a coal plant or something like that. Is that actually a capture, or is that a utilizing that?
It's utilizing. It's utilizing. That's right. So you're just utilizing it one more time before you make the fuel out of it, and then burn it to make additional CO2.
There's still value to that, but it's still ending up.
That's true.
Okay.
In the biomass. That is correct. And so on the surface, this seems like a really easy technology, right? You just grow your algae, you know, laden with all this oil in these open ponds in association with sun and CO2 from these closed ponds or from the flue gases. You take the triglyceride, give that to either a petroleum refinery, you know, Shell, ConocoPhillips, UOP, or a biodiesel producer, and they can make, you know, jet fuel, JP8, biodiesel, green diesel, and maybe others. And then take what's remaining, the carbohydrate and the protein, and either use it for ethanol production, burning it for just power, electricity, or maybe as an animal food feed. So it seems just conceptually really, really easy. But, you know, these 3 steps, you know, algal cultivation, lipid recovery, and fuel production really have a number of barriers and challenges associated with them. Some of these challenges have already been brought up. You know, what is the photobioreactor design that we're going to use? Open, closed, wastewater treatment plant. What are the capital and operating costs associated with each of those scenarios? Temperature control is going to be huge. If you're in a closed bioreactor, you're going to have to somehow cool that bioreactor down if you're growing it in the desert southwest versus an open pond where maybe cooling is not going to be as much of an issue, but it still could be. Saline water chemistries around the country could have a huge effect on the regulation of the lipid pathways in these organisms. How are you going to make that water up that's being evaporated in these large open ponds? And as the water evaporates, you know, if you're using a saline aquifer, that salt concentration is going to get higher and higher, and that may start getting inhibitory for your organism. Growth rate, robust hair. Resistance to invasion is another one. And so if you're growing algae in an open pond and you're trying to maintain what's known as a uni-algal culture, one strain that produces large amounts of oil, what's going to prohibit, some bird from flying over, pooping into your pond, and which contains algae probably, and is going to take over that pond because it has a more aggressive growth rate than your resident algae. Biofouling in closed systems is going to be important, because if you have an organism that has a tendency to biofoul, adhere, biofilms, produce biofilms, adhere to the surfaces, you're going to have to shut everything down, drain it all out, clean it all out, and then start all over again, and that could be very, very expensive. And so these are the sorts of things that people have to start thinking about when they talk about something that seems very simplistic like algal cultivation. We think that probably some of the more challenging, if that's not challenging enough for you, it's how you recover the oil cost effectively. Both dewatering method, that is removing all that water from the algae biomass, and then once you have a slurry of the biomass, how will then you extract that oil from that biomass? You can't use the same sort of crushing methods that you currently use for soybeans, because they're just too small. So you're going to maybe use solvents or something like that. But again, not very green technology. Yes.
That's what I think ASU they say that the preferred method is some kind of chemical extraction, or but it's resulting in some really nasty compound that I can't even pronounce.
Yeah, exactly. Well, I mean, if you're going to be using a solvent, chloroform methanol or something like that, and so your residual feedstock is going to be contaminated with that, and so the chances of being able to sell that as a feedstock for cows or whatever is not going to happen. So those, in our estimation, are really the two most challenging things that we have to overcome. Can you do it? Yes. Can you do it economically? No, not yet. And then, of course, the fuel producers have to deal with a variety of things like, what are the fatty acid profiles and how does that impact the fuel production in terms of the energy density, carbon numbers, cloud point stability, and consistency. Many of the same sort of things that biodiesels producers are facing these days. This was an interesting exercise that we undertook a little while ago. We took a look at what are the number of algae references out there. And the algae references were all the algae references. We're not talking about biofuels from algae. From a period of 1996 to about 2007, you can see that per year, there was less than 50 articles per year total in algae. Yet here is cellulosic ethanol, now a dedicated fuel. All of these obviously started ratcheting up probably about 2003. You can see that that's just going through the roof. This abysmal publication rate really mirrors the type of funding that we currently have out there for academic environments. It's absolutely abysmal in terms of what the academics are putting up. Yes?
These are all peer reviewed?
Yes. Okay. Yeah, these are peer reviewed, yeah. I mean, it's just really, really low. So, who's interested in algal biofuels? This is just a partial list of some of the, probably 200 companies worldwide that have jumped into this really over the last year or two. It's amazing. It's almost like the California gold rush, if you will, and it's almost to that state at this point in time. So here, there's a lot of companies that are really kind of hanging their hat on algal technology. We have, a couple local companies, like Solix here, out of Colorado State University, is working on some interesting generations of closed bioreactors that are actually floating in water. So cooling isn't that big of an issue. So if you've ever been up to CSU, to Solix facilities, you can actually view those sorts of things. There are a couple of companies here that are actually, I'm not sure if I have it on here, there's a company called Alginol, who's actually interested in ethanol production from algae. Not even worried about the oil, so growing up probably like a cyanobacteria, large open ponds, but making ethanol instead. And so DOE has actually taken an interest in that. So, all of these companies using a variety of, some are open ponds and some swear to open ponds. Others think that the only way you're going to do this is closed photobioreactors. Oil industry is also becoming very, very interested in this. And so here are some of the biggies in the oil industry. And so first of all, just to kind of blow our own horn a little bit, NREL is currently working with Chevron in the algae oil to fuels area. We signed this alliance back in October of 2007, and we're well underway with the project with them. Shell announced a deal, a joint venture in Hawaii with University of Hawaii and a small company called HR Biopetroleum, growing in seawater, actually ponds. ConocoPhillips has been working with Tyson to make renewable diesel from these fats and tallows from their meat business. ConocoPhillips, Shell, and Chevron are members of the Colorado Collaboratory's C2B2, Colorado Center for Biofuels and Biorefining. ConocoPhillips just purchased the old facility that was up in Louisville. That's right. They bought a 432-acre facility that by the year 2012 is going to be their major training and renewables fuel facility. Now, being part of C2B2, they just announced that they are also going to be funding $5 million worth of sponsored research within C2B2. And the first projects that they're going to be funding, guess what it is? It's algae. So there's huge algae interest from all these companies. Now BP, they're often in Berkeley with their BP Energy Biosciences Institute, funded to the tune of $500 million over 10 years. That's $50 million a year. I don't know if they have an interest in algae, but I would imagine that there's going to be some interest when you have $50 million a year to spend.
So with all these large corporations doing this, how come we don't have any of the articles being published about it?
Because all of these companies here are interested in proprietary information that you were, and so NREL is kind of in a dilemma, because some view us sometimes as being somewhat academic, but then when we work with the industry, they want to protect that information until it's been patented. So it's a difficult dilemma that we have to work with at NREL sometimes if you're working with companies. So the oil industry can obviously take the oils from algae and put it through either their distillate hydrotreater, which is going to give you a green diesel or a jet fuel, or put it through one of their crackers and reduce the size of those carbon links and produce something that's much more akin to the size of a green gasoline or olefins. There's also growing interest by the jet fuel end users. So down this list here, you can see that the engine manufacturers like Pratt & Whitney, Boeing, Air France, KLM, JetBlue, Airbus, Honeywell, and New Zealand Air are all very interested in the potential of using algae as a feedstock for their jet fuel, because you can actually make a fairly credible commercial or military use jet fuel from any of the terrestrial vegetable oils, and the thought is that you can also do this from algal oil. The Department of Defense has also gotten very, very interested in this. That's because if you take a look at their fuel consumption, it basically, jet fuel is 71% of their fuel consumption. And so, they use basically about 5 billion gallons of jet fuel a year. DARPA, kind of the research arm, the very risky research arm of DoD, in 2006 put out a solicitation to prove technical viability of making JP8 from oil-rich crops. These were the awardees. These were really mostly looking at terrestrial kind of crops, but the initial data that came back so surprised the DARPA that they have now put another solicitation in really focusing on algal biofuels. And so here is, this is UOP's data whereby they're comparing a JP8 spec with a bio-JP8 spec that they use via their hydrotreating process. And you can see when you look at freeze points, flash points, and density, they're pretty spot on, except for maybe adding back a little bit of more aromatics here. But again, this piece of information has so excited DARPA that they have jumped into the algal realm with both feet. And so this is the second DARPA announcement that was announced last year. Basically, this is the process for making affordable and efficient production of JP8 from algal oil. They did put in a cellulosic materials, but frankly, I don't think any one of those won out. There have been 2 awards, not officially announced yet, because there's a little bit of, there's a disgruntled team that wasn't chosen is actually contesting the awards. But 2 teams, one led by General Atomics and the other by SAIC. Both companies that are well tuned to doing work with the U.S. government. But these are large, multi-party teams that are assembled here. Investments are also heating up in this area. Venture capital invested about $280 million in advanced biofuels in the second quarter of 2008. $84 million of it went to algal biomass. Sapphire Energy, which is a small company out of California, and till now really nobody had heard about it, but amazing, $50 million was raised to fund this one company's work. Aurora Biofuels out in California, Berkeley area, raised about $20 million. And then here's that company, Alginol, which makes ethanol from phototrophic algae. This is almost too incredible to believe. Announces $850 million investment from a Mexico company called Biofields to produce ethanol from microalgae. So you can see it really is almost akin to a gold rush where all this money is starting to flood in. Now there's also no shortage of ideas with regard to cultivation. I'm going to just show you a couple here. Obviously PetroSun and Live Fuels and Aurora Biofuels, those just name a few, are very interested only in looking at open ponds because they maybe looked at their numbers and they say the only way to make this commercialized is with an open pond. But then there are companies like Valcent down in Texas who are growing their algae in these kind of vertically hung plastic bags where the algae is actually kind of being pumped through here, and then through a pump, and then back again. Okay, it's really kind of amazing. Green Fuel has been in this area probably for the last 3 years or so. They're kind of one of the initial ones that have gone through a lot of different generations. This was, can't remember what generation photobioreactor this was, but this down in the deserts of Arizona is probably more akin to what their newest generation is. But Solazyme, a California company, they're growing their algae not phototrophically but heterotrophically. They're growing it in fermenters, for God's sakes, with sugars, and so making oil from algae that way. And so, obviously they must think that business model can work in the future. A2VE carbon capture, which is still kind of a virtual company here in Colorado, is looking at generating these large farms with these units here, which can hold like 300,000 gallons of water, and each one of them growing an algae. And so if one becomes contaminated, no big deal, you got all these others. So they're really not connected. So these are just some ideas that people are thinking of. And then additional ones. These are some vertically hung bags at Arizona State from Quang Hu's lab. In Israel, they're looking at some of these flat panels. Israel is also looking at these long tubular structures that you see here full of algae. And then Utah State University is taking advantage of some technology developed at Oak Ridge National Labs. And again, this has got to be pretty expensive. I mean, whereby you're taking sunlight, concentrating it down these tubes, if you will, these photo optics here, in which case down here is where you're actually having the algae grow. How you can ever do that economically, I don't know, but people are pursuing these very interesting ideas these days. There's also no shortage of public demonstrations, or sometimes, as I refer to them, as stunts. Here's Sir Richard Branson, from Virgin Atlantic. Back in February, they actually flew from London to Amsterdam using a biofuel blend of Babasu oil. Does anybody know what the Babasu is? And coconut oil mixed with kerosene. So here's Richard Branson kind of throwing a coconut up in the air. He's always in the limelight. And then back in January, Solazyme, remember they're the ones growing in these fermenters in the dark. Basically made enough biodiesel from the oil to actually power a Mercedes, and very highly visible, drove that through Park City, Utah during the Sundance Film Festival. Okay, so that's kind of a long-winded introduction. I'm going to now go very quickly through what we at NREL have been doing that is now beyond the aquatic species program. It really all started probably only a couple years ago. At the time, NREL was doing absolutely no work in the algal oil area. However, at the time, we had a very robust algal biohydrogen system led by Mike Seibert, who's here in the audience. At the time, we wanted to explore what is the complex landscape of algae-based biofuels, determine the most promising algae to biofuel scenarios that was complementary to what we were doing in biohydrogen, And then to next, strategically position NREL to become competitive again in the broader arena of algal-based biofuels. And then lastly, to seek funding for the most likely biofuel scenario. And after this analysis, it really came down to the use of these higher energy oils. Because when you compare the energy content of oil, carbohydrate, and protein, lipids win hands down every single time. The cornerstone of what we're currently doing at NREL is this Chevron Algae CRADA, which stands for Cooperative Research and Development Agreement, signed back in October. The goal here, and again, I can't talk about too much about this, otherwise I'd have to kill y'all, is to really, for us to focus in on the biology, to identify and develop algae strains by breeding or by whatever means, to make algae that are economically harvested and processed into finished transportation fuels. So we work on the biology, and then ultimately the oils produced by these algae would then be given to Chevron and would use their proprietary technologies to make a variety of biofuels. We've also been working with the Air Force Office of Scientific Research, and I know that Mike Seibert has been funded by them for quite a while, and so they have an interest in basic biology, and so that's why they've been funding biohydrogen research, but they also had an interest now in algae to jet fuel. And so we've been working with them for a couple of years now. They're actually funding a few academics that complement very nicely what we're doing at NREL. And then back in February of 2008, we actually held an algae workshop for them where we brought in largely academics, because they're the ones that are focusing in on the basic biology of these organisms. And so this is just to give you a sense, and you may recognize some of these names on this list. These are just some of the workshop speakers that we organized. We talked about what had gone before the aquatic species program. We talked about bioprospecting, role of culture collections. We had Jerry Brand, who's the culture curator from UTEX. Ginger Armbruster from University of Washington talked about sequencing of algal genomes. There's only a handful of algal genomes that have been processed so far. If we're going to make a valiant attempt at biofuels from algae, we're going to need to sequence more algal genomes. Steve Mayfield from Scripps talked about algal model systems and genetic tools that they've developed. General lipid biochemistry, Wayne Rykoff, who actually I think was a graduate student in Christoph Benning's lab from MSU, looking at triglyceride pathways, identifying those, what's the regulation. Ami Ben Amatz from Israel is an expert at growing algae in large-scale open ponds and all the things that are required to do that. Jonathan Trent at NASA Ames gave us a very interesting scenario where he talked about the Netherlands' interest in potentially growing algae in the ocean. So there are some bits and pieces of interest here, but again, that's still a long way from reality. And then the next day we had Trig Lindquist from California Poly talk about algae and wastewater treatment, again taking advantage of the cheap nutrients. Quang Hu at Arizona State, closed photobioreactors. Sally Sun, who's part of the Arizona Public Service, working in conjunction with Green Fuels, trying to produce oil in Arizona. Mark Hildebrand developing genetic tools for predominantly diatoms. Chuck Dismukes at Princeton, algal photosynthesis. Byron Wood from Utah State, a little bit about technico-economic analysis. It's all going to come down to the cost. And then lastly, we had a lawyer, of all things, come in and talk to us about policy and regulatory issues, because if you're going to be growing large amounts of algae, let's say even in open ponds, and even more so with a genetically modified organism, you know that somebody at some point is going to come along and say, we're going to regulate you. So getting in front of those people now rather than later is going to be absolutely critical. Trying to contain some of these organisms and making sure what are the impacts if this organism that you're growing on literally millions of square acres gets out into the aquatic environment, whether it be a stream or pond. What's the impact on that effect? All of the presentations from this workshop, the abstracts, the agenda can be found at the NREL.gov biomass website. So all you have to do is go http www.nrel.gov/biomass. When you get to that site, on the right-hand side at the bottom is a link for all of these presentations for all of the abstracts, etc. And I think they were very, very useful because we're using that information currently to actually generate that roadmap that's going to help, hopefully, the Air Force Office of Scientific Research develop future plans for algae. The other areas that we've been actually blessed with is that NREL has actually seen value in this as a potential biofuel in the future. And so they have funded us. Oh, the animation didn't come out. Oh, what a bummer. There was some animation here. I'm not sure what happened here. But this is a fluorescence-activated cell sorter, also sometimes referred to as a fax machine, not to be confused with a document sender. This instrument has been used routinely in the medical research environment to actually sort various eukaryotic cells, T cells, B cells, things like that. We're using it here at NREL to actually sort, in a high-speed, rapid methodology, sort algal cells. And so you can imagine going out to the environment, collecting a water sample, which is hopefully full of algae, and then using, first of all, the chlorophyll fluorescence that we have here, and then using this cell sorter to actually distribute single droplets with single cells into either a tube or either into a 96-well plate. So in a very quick way, you can develop 96 pure cultures of algae in literally a minute. Okay? Remember the aquatic species days, it took them a whole 5 years to accumulate very laboriously, going out and getting the water samples, plating those out, waiting 2 weeks for them to grow, picking a colony, streaking it out again. Hugely laborious. This is, pardon me? No, not at this point. So if you wanted an exenic culture, you would then probably resort and then test whether or not these wells are contaminated with bacteria or something like that. So the chances are at this point they're not exenic, but you can then make them exenic using this instrument. We've also gotten some monies to start freezing some of these things down, because as we start collecting a lot of algal cultures from the local environment or other places, we're going to want to freeze those. And so I've been in contact with Jerry Brand at University of Texas, and this is basically the instruments that they use down there routinely. They swear by these instruments, the MVE units here, minus 190 degrees centigrade in the vapor phase here, really minimizing damage to algae at low temperature. It's going to provide long-term cell viability for most, not all algae can be frozen, and preserve any intellectual property that may be associated with these algae that we collect. In addition to that, we've gotten some monies to renovate our greenhouse. Yes, we do have a greenhouse at NREL at the top floor of the FTLB. The greenhouse is very poorly laid out right now. This is an enclosed, temperature-controlled room, which is not much use, and so we're going to demolish that and push that over to the end over here and construct a space which is really dedicated only for algal bioreactors. It's also going to liberate a lot more floor space so that the folks who are actually growing things like poplar trees for looking at chemical composition, etc., in terms of cellulitic conversion, they'd have a lot more room on the floor.
Hey, Al?
Yes?
Was that old raceway paddle wheel thing accessed eventually, or?
Yes, and I don't know where it went. That's what Jonathan is referring to is on the floor, right about here. They actually had a kind of, maybe a 10, 12-foot long raceway that was actually growing algae using the sunlight coming through the. Whatever happened to that 12 years ago, I have no idea, because I've been only at NREL.
It's still there when I was an intern about 2000, just sitting there gathering dust.
It's gone, and I have no idea where it is.
It's pretty funny.
Yeah. This is a project that we're also currently pursuing with C2B2. I mentioned this is the Colorado Coal Laboratory. So last year they handed out their first, their seed grants, and I was able to get one of these. I'm actually working with Matt Pozowitz and a graduate student, Lee Elliott, on this. Basically, we're taking advantage of the high throughput nature of the fax sorter, the robotics that we have at NREL, to really establish a bioenergy focused microalgae strain collection using these rapid high throughput techniques. Again, going out to various environments, sampling from creeks and ponds and ponds that have high, low pH, ponds that have high, low salinity, etc. Really trying to get a handle on what are some of the organisms that are in there, but more with a bioenergy focus. Yes?
Is that research bound by your funding through the Air Force, or is this information that's going to be shared with industry? Well,
this is quite interesting, because C2B2 has about almost 30 sponsors now. Okay? And the sponsors, every year, they pay a sponsor fee. And it's these sponsor fees that actually fund these seed grants. Now, any IP that is generated from these seed grants can be shared amongst the sponsors. Okay? Now, it's possible that at some point that these strains that we developed here from this project could be available publicly. But again, I think the sponsors will have first crack at maybe whatever is developed from this. But the nice thing is that these 30 companies can actually share in the IP here. We've been routinely using Nile Red. Nile Red is a lipid stain that people have been using for quite a while to stain lipids in algae, because for the most part it's kind of specific for neutral lipids. It has a pretty good Stokes shift. We've been recently using, however, this dye called Bodipi D3922. In the literature, they say it's a little bit more specific for lipid droplets, that is neutral lipids, than Nile Red, although both are very, very useful. And using, the Bodipi strain, this is the type of fluorescence data that you get. So this here is a diatom called Phaeodactylum tricornutum. Here you can see this is just the staining pattern that you would get for chlorophyll, so that's representing chlorophyll. But you can easily see these large green fluorescent droplets in here, which we can only assume is neutral lipid that can be used for biofuels production. So you see minimal fluorescence. And so this is a fluorescence range here anywhere from 100 to 10,000 to 100,000. Okay. So when you stain this sample with Bodipi, all of a sudden you see this huge spike in fluorescence out around this area here. This channel here just shows you that you have different cell sizes, anywhere from very small to very large. And when you stain this, you can see that that size population is still there, but it is now much more fluorescent. Okay? You can now layer on top of that chlorophyll fluorescence and emission. And now you can gate a population of cells that are both fluorescence due to Bodipi and fluorescence due to chlorophyll, and actually sort those populations into 96 well plates. And so now we're not just sorting based on chlorophyll, that is, it's just an algae, but it's an algae that in theory should be producing a neutral lipid, which can be produced in oil. So can you imagine what the aquatic species program would do if they had an instrument like this that would actually pre-sort for them organisms that may have huge lipid levels? We also have internally funded money at NREL to start developing analytical techniques for assessing lipid production in algae. So Ed Wolfram, who is an expert in near-infrared spectroscopy and has applied that technology to determine content in cellulosic feedstocks like glycan, glucan, xylan, lignin, etc., is going to be applying those same sort of techniques to algae. And so here you basically do wet chemistry to analyze a variety of different organisms that are producing these large globules of oil here. And then using these as data points, you can actually develop a model to determine how much oil is actually being produced in these organisms just using a simple, near-IR spectroscopy. So that actually just started just a few weeks ago. So we have high hopes for this project. We have another project which is pending. This is not funded yet, but Phil Piankos and Maria Gerardi at NREL are going to be collaborating and using some digital gene expression. That is a fancy word for some of the Celexa or 454 sequencing that we currently have available to us, using it to basically sequence cDNA libraries that are produced from mRNA to give you a handle on what are the RNAs that are transcribed during growth under different conditions. And so under conditions where they're not making oil or they are making oil. What are the genes that are upregulated or downregulated to that? And so hopefully this will be funded. This could be quite substantial because we've asked for about $500,000 over 2 years to do this project. We also have a pending project with Israel right now. I mentioned Ami Ben-Amatz, who was at the AFOSR workshop. We have a tentative working plan with them whereby they produce algae in these large open ponds in Israel off the Mediterranean coast. They grow a number of different algal species in here, greens, diatoms, depends on the time of the year. We are going to be using the biomass that they generate to develop cost-effective, or try to develop cost-effective methods of extracting these oils, potentially even using some of our thermochemical gasification techniques for gasifying the residual biomass to see if that's cost-effective. And then AMI is going to be using their funds to try to, improve, more cost-effective processes and harvesting processes of this algae. So this is pending as well. But this, interestingly, is through the DOE. There's also a new effort afoot, used to be called the Southwest Center for Sustainable Algae and R&D, and I know they've gone through a couple name changes. They're a consortia, they're a center, not really sure right now, but basically back in May we got together a group of folks, Sandia, NREL, OBP was represented at the DOE Office of Biomass Program, a company called Carbon Capture, Cargill, a number of universities, Mexico State, Arizona State, CSM was represented, Matt Posowitz was there. Basically a working workshop to try to organize this more kind of regionally based consortium. Phase 2 in 09 is really to develop a 10-year comprehensive roadmap or research plan, and we think that actually may be carried out through the DOE. And so DOE may actually do this for us, but then for the next 10 years they have a very ambitious goal, whether it be attained or not, of attracting $1 billion worth of funding, whether it be through federal sources or maybe even dedicated industrial sources to really try to carry this through. But again, more regionally located, New Mexico, New Mexico, Arizona, Texas, Hawaii. I guess that's the southwest part. That's the west part, southwest. So we'll see kind of where this goes. We've also had some outreach activities. My colleagues at Arizona State recently published a review article in a special issue of the Plant Journal, Harnessing Plant Biomass for Biofuels and Biomaterials. And so we wrote a review article on microalgal oils as feedstocks for biofuel production. So this was the May issue of the Plant Journal. And then my Colorado State University colleagues and I are currently writing a paper trying to limit what is the theoretical maximum of oil production from a typical algae. There are companies out there who claim sometimes to say that we can make 100,000 gallons per acre per year. Sorry, but you just violated the first 2 laws of thermodynamics. It ain't gonna happen, okay? And so what we're trying to do here is, okay, what is the theoretical, which will never be attainable because it assumes 100% efficiency, et cetera, but what is a more practical production for algae? And we're thinking it's gonna be probably in about a 5,000 to 6,000 gallon time frame. We've also, because we're such a high visibility laboratory, we've been quoted in NPR, CNN News, Wall Street Journal, CNN Money. I mean, I'm getting calls right and left from companies who want to do interviews, and so this takes up a large part of my time as well. But the thing that I do here is try to give them a realistic expectation, because they get on the line and they say, oh, we've talked to this company, we've talked to this company, it's exciting, all this. I mean, we're talking commercialization next year, right? Wrong. So I tend to, give them, kind of deflate them a little bit, but I give them more or less a realistic picture of where we are currently, but where we need to be in the future. In 2007, in December, the President signed the 2007 Energy Independence and Security Act, which provides, increasing renewable energies that decrease gas house emissions, providing 36 billion gallons of renewable fuels by the year, 22 of which, some of this is corn ethanol, and a lot more cellulosic ethanol, and some other alternative fuels as well. Biodiesel's in there, renewable diesels in there as well. But if you go to Section 228 of that Security Act, look, there is language, believe it or not, in algae, requiring the Secretary of Energy to develop a report delivered to Congress on the feasibility of using microalgae as a feedstock for biofuel production. That's huge, because in the past, DOE has really been focused in on cellulosic ethanol. Okay, it's going to be a transitional fuel, but this starts really, giving us hope that in fact microalgae for fuel production may ultimately, there may be much more funds coming in this area. Yes?
Did that report happen?
It did, and I was involved in it. And it's been delivered, but not to Congress yet. So they haven't delivered it to Congress within 90 days, but that's the government. You know, they always put these, you know, grandiose sorts of things. But can you imagine? We wrote that report in 57 days. It was huge. I mean, covering all aspects of this. I mean, it was huge. And so the hope is that this will make it to Congress at some point. And you know, personally, I think it's probably going to be one of the best reports that nobody ever will read. Maybe except for some staffer in Congress. But no, the hope is that this is really going to make some splash in terms of the microalgae biofuels.
So can we request a copy from you?
Pardon me?
Can we request a copy?
I can't. It's right. It's basically DOE's report. Okay, we got it together for them, and until they release it or it goes to Congress and Congress releases it, we really can't do that. Unfortunately, I'm sorry. So let me just finish up here. The promises. Okay, technical feasibility has been demonstrated. We can make oil from algae using sunlight. We can do it. Okay. The goal here is whether or not we can do it economically, okay? So we can use it to make biodiesel and these other refinery feedstocks. It doesn't impair, you know, food production or water land resources. And it really has a potential to supply a significant percentage of U.S. fuel demand. The challenges are many. Basic algal biology, I think, is still huge. Process research, production of the entire integrated process that is cost effective, so economic analysis all along the way, the research is going to be absolutely critical. And then what are the environmental and social issues of growing these large algal ponds? And so I thought I'd finish up with this right here. This is interesting. You can actually go to the web, and there is somebody who is marketing this book, Making Algae Biodiesel at Home. Okay, I mean, I can see how people can produce biodiesel at home from waste oils and things like that, but this guy is telling you that you can make algae biodiesel at home. He'll only charge you $99 for the price of this book. And so we've actually thought about ordering this book just to see kind of what's in it and how they're going to grow. I think they're growing these things in aquariums, so can you imagine making fuels from algae?
Oh, you got to stick around. He's coming. Actually, he's next.
Is he really?
No, I'm just joking.
Well, I'd like to talk to him, actually. You know, I would love to talk to him. So let me just finish up with this. People who know me that I'm a huge Calvin and Hobbes fan, and this was the very last comic strip that was produced by Calvin and Hobbes, Watterson. Basically, you know, Calvin and Hobbes are going tobogganing, you know, snowed last night, everything familiar disappeared, world looks brand new, fresh, clean start slate, white paper to draw on, day full of possibilities, magical world, Hobbes' old buddy. Let's go exploring. I really think that in terms of the algal biofuels area, I think this is where we are right now. We're really exploring the possibilities of the algal realm right now. And so I think this is really kind of where we're at. So with that, let me just acknowledge my collaborators. These are some of the algal folks at NREL, Eric Jarvis and let's see, where is he? Eric and Nick Daigle, sorry, are both from the Aquatic Species Day, so they were at NREL during the Aquatic Species Days. These are my external collaborators, people who helped us with the report, Matt Poswitz, who we're collaborating with, Lee Elliott, my Arizona State collaborators, Who and Milt Sommerfeld, CSU, Brian Wilson at CSU Solix, Walt Kazumbo at AFOSR, Ben Amatz, and our work currently with Sandia and Brent. So with that, I'll thank you for your attention, and we'll answer any questions you may have.
Some of what I've read indicates that getting the high yields of oil from algae requires stressing them, probably by withholding nutrients.
Yes,
The trade-off is that reduces reproduction rates, so that the net yield is better if you don't do that, and you just take the lower percentage yield.
Right.
And what's your experience and opinion on that?
I don't really know. We are more focused in on finding at the molecular biology level what those triggers are and how oil accumulation is actually regulated. Wouldn't it be nice if you could get a strain of algae that you somehow bred or genetically engineered, that as it was growing, it was fooled into thinking that it was being stressed, whether nitrogen was being removed or some other stress, but at the same time it was accumulating those lipids for a rainy day, if you will. So I think that it's probably equally divided. Some people think, yeah, we'll just grow it, and whatever lipid it produces, we'll get. Or no, we're actually going to dedicate a pond, if you will, to stressing it, and then we'll isolate it.
Grow it first and then
Right, and I think there's people in both camps. Yeah. Yes.
Is it possible to do a tour of the laboratory, or?
Yeah, actually, if I'm not mistaken, because we're so busy, it's very difficult to just give one-off tours. But I think the third. And Mike left, but I think the third Thursday of every month there is a tour that is given at NREL, and you can actually tour the FDLB facility. So call NREL, or if you want to give me your card, I can send you that information. And so once a month we have large groups that come in and they can tour bits and pieces of NREL, one being the FDLB lab.
You might want to mention, Aldo, there's a little bit of lag for international folks.
A little bit, but if we get their names and contact information well beforehand, it shouldn't be a problem.
Yeah, 2 weeks, right?
Yeah, at least. Yeah, exactly.
Thank you.
Yeah, you're welcome.
Exactly.
Yes.
About a mile
Extraction.
Tough area. Very tough area. Yeah, it's like I said, it's.
Anything new?
No, I mean, there are a lot of people who are, like as I mentioned, doing solvent extraction. Other people who are looking at various, chemical ways, mechanical ways of trying to disrupt the algae. But I'm from Missouri. If you have something that's economical, show me. But a lot of these small companies are playing their cards like this. So you're like, well, but they go out there and they claim all this, and then you ask them, well, what are your economics? What are the costs? Well, we can't really share that with you. Okay, so I'm supposed to believe you, right? Yes.
I have a hard time finding anyone that is willing to let the public look at their site, like even Solix. They won't let anybody come and visit.
I would agree with
See
you.
what they're doing?
Yeah.
Do you know of anybody that's willing to let us see?
Yeah, I think there are some that are a little bit more open, but again, what they may be showing you is maybe their first generation. They're already on their fifth generation of bioreactors, and so that's
I'd be happy with that.
Yeah, well, I mean, Solix might show you, some of their first incarnations, which are basically these large plastic bags that just kind of laid on the ground, and a roller just kind of came over them to mix them back and forth. But they've moved on, since then. So, no, you're absolutely right. It's very difficult to get into any of these places to see what they're doing. Yes.
You mentioned one particular study which was still in the can waiting for some money. I imagine you have more projects that are even cooler than that might need a lot of money. And I'm wondering, is appropriations for NREL just looking for big companies, or who in the government can we get to partner with NREL so that we can actually get the research done?
Well, and it's not just NREL. I have a feeling that as DOE starts funding some of this, they are going to be putting out competitive bids. I think that, I think the days of where NREL just gets a boatload of money to do this work, I think is over. I think it's going to be very competitive. So you'll be seeing. Granted, there'll be more solicitations that are geared more toward, let's say, national labs. There will be solicitations geared toward universities as well. In fact, I think one that closed a little while ago, a university solicitation for algae. I think those are actually currently just reviewed here at the NREL site for funding. So funding is, I think, on the upswing, but it's not here yet. Yes?
Let's say you had a sewer plant available right now and it was in the process of rebuilding its infrastructure. Who would be the best company to talk to about some kind of joint venture, at least designing the tanks?
Oh, designing the tanks. Oh, that's,
so it's the opportunity not lost, later implement.
Yeah, I mean, there are a number of venture capital firms who are interested in this area, but they're just interested in those companies who already have something designed. So what about starting your own company? That's a viable feasibility. I mean,
the money's already there.
Yeah.
Work's being done.
Yeah. Because we don't do a lot of work with those venture capital firms. I really wouldn't know at this point. Sorry. Yes.
The efficiency of algae turning a photon is about...
It's maybe only about, what, 5 or 6% or something like that. The photosynthetic efficiency of, now photosynthetic efficiency of algae is still considerably higher than most terrestrial crops. And in fact, one of the most productive terrestrial crops we have is sugarcane, and which is only also about 5 or 6%, or maybe higher, maybe 10, I'm not sure. But algae, and so that's another reason that people are looking at algae, because of the higher photosynthetic capabilities. Joe,
yeah, I had a comment. I thought you were kind of very kind to a lot of these algae startups, because they, a lot of them have focused on the bioreactors, and they've built very expensive tubular bioreactors that have brought in a lot of venture capital. But no matter how you grow a tomato, there's not going to be a whole lot of lipids there until you find a canola. And we can't taste algae for oil. We can't see. We don't have senses. And only with the kind of technology that NREL is utilizing and other research groups are utilizing, we're going to find the right organism. Now, these techniques in this time frame isn't available to these venture capital companies. But they are doing a good job of acquiring venture capital. So I guess I'm jaded because I'm a phycologist and I'm in academia. But it seems like I wouldn't look to any of these companies to really make these breakthroughs. I'd really look to the people who are looking at the hard questions that are going to move this technology forward. I mean, would you agree with me on that?
My feelings, exactly. Now, being a part of a national lab, we have to be very careful where we tread. First of all, we can't lobby. And then when people call us up and say, what do you think of the technology of this one company? We really can't make any predictions. We just basically call it as we see it, which is we think that both applied and basic research on the biology needs to take place, and that it could be several years, 5 to 10 years, before we get a commercial entity out of this. Yes?
Yes. If this becomes cost effective, in the spirit of this conference, what do you think, how do you foresee the accessibility to small producers or co-ops or something like that?
Yeah, I think that's a concern. And the reason I'm saying this is that look at the oil companies. Look at the infrastructure that they have here. They know what to do with oil. And so if all of a sudden there's 30 billion gallons of algal oil put on the system, you'll hear this huge sucking noise to the refineries, because they have the infrastructure. Now, does that mean that the small co-ops and the like are doomed? No, I think there's probably at that point going to be plenty to go around. But that's why these oil companies are getting involved here, because they can deal with that kind of stuff. But I think there's still going to be plenty to go around for co-ops.
You don't think they're going to tie that up in patents so bad that you look at it and
Well, I don't know. That's a good question. I think there's so many people out there right now that frankly, there's not going to be one winner. There's not going to be one open pond. There's not going to be one closed bioreactor. There's not going to be one organism. I think there's going to be a lot of different technologies that are going to be making up this 30 billion or more gallons a year. It's not going to be one. I guarantee it's that.
Yeah, along the same lines as that, I think in small-scale biodiesel, at least traditional biodiesel production has brought a lot of new innovative technologies to large-scale biodiesel, in fact, which is pretty impressive, and I think shows the value of grassroots technology building, which is impressive. And so my question is, there's lots of folks out there who want to get their hands in this. What's sort of the best place for a small-scale person to try to take part in advancing this technology in a way that a small-scale person can also take advantage of it? So where can you sort of get your hands dirty in algae?
It's like that scum that's in your hand there, yeah.
There was that book here showing up.
There's, for example, there's an algae for oil group on Yahoo groups, in which a lot of talk goes back and forth, and people constantly refer to, well, look, Solzheim said this, and look, these guys said this, and there's maybe one or two guys with a bag in the back of their house. But besides that, no one's really doing anything serious. So how can we start to do things on a small scale?
It's possible. Obviously, you'll need a laboratory, but I think you need more kind of sophisticated things to make oil from algae a reality. I mean, it's not just taking the oil and mixing it with methanol and base and then separating it and washing it and away you go. It's going to be a little bit more difficult than that. And so it's because you're dealing with biology at this point. You're not dealing with the product of biology, which is oil. And so that, I think, is difficult for most small entrepreneurs to get their hands on because they don't have, first of all, maybe they don't have the biology experience of growing things. I mean, I got a call from a company that were mostly chemical engineers that didn't know, boo about growing algae, and so that's going to take some time. And so, it's not an easy thing because you're trying to harness biology. And on a small scale, it's tough to do without sophisticated equipment. Yes?
Getting to the finish line first. Corn, cellulosic.
Yes.
Soy.
Yes.
Biodiesel, algae.
Right.
Who do you think is going to get to the finish line?
Well, DOE is certainly focused on cellulosic. Okay. We think that over the next 10 years or so, cornstarch ethanol is going to peak out probably about 15 billion gallons. We're making 7-8 billion right now. That's going to peak out without putting adverse pressure on food price or anything like that. But by the year 2030, granted I'll be retired by then, but DOE's goal is to make 60 billion gallons of ethanol that they could use for, and you saw the 2022 goals. Now that's going to be a mixture of cellulosic ethanol and biodiesel and all these renewables. So I think cellulosic ethanol is not too far away. I think we can see it at the light at the end of the tunnel because DOE has been pumping in millions of dollars in basic research trying to develop better fermentation organisms, better enzymes to do the saccharification, pilot plants. They're building pilot plants in association with companies, 10% scale, commercial scale, and so I think that's going to become a reality. But at that point, ethanol I think is going to be a transitional fuel because if you can do cellulosic ethanol, you can do butanol, okay, which is a much better fuel than ethanol. Although, I didn't say.
What if nobody wants that?
Yes.
The average power plant in the U.S. is about 350 megawatts, I believe. And I'm just curious if you're partnering at your power plant using their CO2 in the exhaust stream, how many gallons per year of a algae field?
Yeah, that's a good question. I don't know. In fact, maybe going back to the aquatic species program, we may be able to find out. But so oil companies are obviously interested in this. Energy companies are now starting to become interested in this because they have a coal-fired power plant over here with some nasty flue gas, natural gas burning power plant over here. Flue gas is a little bit more pure, and so these people are now interested in maybe producing biomass for production of electricity. Maybe they're not too interested in biofuels, but if we can use the algae to sequester the CO2 that we produce, then we appear a lot more greener to the public. And so,
actually, dollar amount now, carbon credits.
Right, carbon credits.
Yes,
and carbon credits are going to be used, and so you'll find companies like Excel and some of these other companies start becoming. Let's start looking at algae biomass, not for biofuels, but for sequestration and carbon credits. Yeah. Yes.
We've been sitting here saying, algae biodiesel, biodiesel conference, but algal-based ethanol. What do you think about the future of that? The chemical refining steps are a lot less intense.
You're correct. You're absolutely correct. In fact, you don't have to worry about harvesting. You don't have to worry about extraction. You just basically have to remove the algae or from the water or the beer, if you will, and then somehow purify, distill, or whatever have you, and then you have the ethanol. So I'm actually going to be visiting Alginol in Berlin and in Spain in August, and so it'll be interesting.
And there is one innovator who supposedly drove his car 70 right past NREL on biobutanol from algae he harvested from.
A number of years ago, yes, I saw he drove across country,
yeah,
right?
That's right.
Across country using
he posts regularly on that Yahoo group.
Like a 1998 Oldsmobile or something like that, and put just straight butanol. Straight butanol and drove right across the country.
You got to pay a little tax on that, but
yeah,
pull it out of a still in your backyard.
Yes.
Do you know of any research utilizing electrical current to extract oil from algae?
I think there may be some people down in Texas who are actually doing that. They're providing some sort of electric charge that kind of stresses the algae, kind of pulls them apart a little bit, and then quickly releases them again, and I think that somehow is able to release the oil. And so I think there are some people that are looking at that. Again, can you do this cost effectively when integrated in the entire process? Yes?
We've been talking about cost effectiveness and based on the demand for fuel. And I heard that XL Renewables, I haven't seen it but down in Casa Grande, that they were doing like kind of a farm scale model where they're looking at the algae oil being produced as a byproduct from creating algae as feedstock for their farm system.
Right.
And the sort of the a part of that, I guess part of my question would be looking at, you know, how valuable is the algae oil being produced right now when you look at like cosmetics or food for animals, which is really, you know, kind of a huge demand on waste oil streams, which are in demand for biodiesel production as well.
The algal oil is really not worth that much. In fact, if you have an algae that's producing your biofuel oil and some other high-value component, it's that becomes now the driver. And it's like, oh boy, what are we going to do with this? Oh, I guess I'll make biofuels from it. So if you can make, you know, a higher commodity chemical or intermediate from the organism, and then combine it with maybe the carbohydrates, make ethanol out of it, make the protein being used for feed, then it starts becoming a lot more economical. But you're right. I mean, most large-scale algae growers these days, they're producing nutraceuticals. They're producing these long-chain polyunsaturated fatty acids for human consumption. You know, baby formula, you know, omega-3 fatty acids, those sorts of things. They grow algae in large open ponds. You're not, well, actually, Martech is actually growing some of theirs in closed photobioreactors as well in the dark. Why? Because they can take the hit, because the stuff that they're making is, I don't know how many hundreds of dollars a kilogram or whatever. Yes?
I won't quote you on this, but what's your personal preference, open pond or bioreactor?
Oh man, that's you know, being at NREL and reading the aquatic species program and not having the information from all these small companies that are doing closed photobioreactors, I would say this has to be open pond, you know, at some point. Now, again, you know, make believe out of me, show me that you can do this cost effectively with a really cool photobioreactor system. And we'll see hybrids between an open pond and a closed pond. So, yes. Those most oftentimes those are probably just water, you know, sources that have algae blooms, you know, and I'm not sure if those are dedicated, whether those are maybe, and those could be just due for nutraceutical applications. But I think more and more we'll start seeing those ponds, green ponds, where people are actually growing them for biofuels applications. In fact, there are ponds you can see from Google Earth, from petroalgae, you know, you can actually see these oval tracks. So again, I can't comment. I don't really know what those ponds are. Yeah.
Well, just one last comment. I hope that like NREL and other government groups and academic groups are interested in working with the public. You know, the public, I think, at large may not be the greatest sort of source of technology, but it is sort of one source in the same sense that there's 200 companies out there. There's definitely there's a group of people who are pretty dedicated and interested, and it's surprising the creativity that can come out of that.
Yeah.
So I would certainly like to.
Yeah.
I certainly hope that you would look into providing some or initiative or just general interaction more with folks like this.
Right. You know, in later this year, DOE is going to be, and this is not widely known yet, but it will soon be. DOE is going to be having a workshop on algae. And so, who are the stakeholders that should be involved? You know, obviously academia, obviously national labs, obviously companies, but you know, what are these? Who are these grassroots groups, and who would be their representative? Because we can only invite so many people. So who might represent? So if you can give me some guidance there, I'd love to have that information. I really would. So the more people we have thinking about this, the quicker it's going to take place, as opposed to letting this small company over here, which is swimming in money because it's some venture capital firm. So the more people we get involved, the better.
Yeah.
VC money is good.
But VC money is good. I'm not saying it's bad. There's a lot of development. But this, I think this is bigger than one small company. This is really going to take a multitude of companies, national labs, and universities, etc.
I'm sorry about VC.
Yeah, VC money is good. but it's just a part of the picture. It's part of the picture.
Thank you very much.
Yeah, thanks. You know what I can do is, when I get back home, I will see what I can, most of it I can make a PDF and send you a PDF.